Welcome to another broadcast from mindyourhealthcast.com. As you will recall, that if you watched our last broadcast, that we introduced some what I consider to be up-to-date information regarding DNA. Now, I called it, title-wise, DNA from Mechanic to Ghost, and you'll see what I mean by the ghost part of it by the time we get finished, and I'm not sure if we're going to make it this time through all of this or not. It seems that just in the last several years some of the biggest strides have been taken, and at the very beginning, as I mentioned before, it seemed pretty simplistic. And it seems that that's the way it usually is when we step into a new threshold. At the beginning, we know there's something going on, and we start looking, and we finally find something that seems to explain what's going on. And wow, we jump into it, and we come up with a very simplistic principle. And the funny thing about it is that in short order, the little, what I call genies or trolls come out from under the bridge. The genies make an appearance, and all of a sudden you find, now wait a minute, it's not quite as simple as we first thought. Here's the general principle or role, but then there's all these little fine-tunings that, as they used to say, it's an old saying that the devil's in the details. It's all of a sudden these little details that start popping up that begin to expand the knowledge base, and we begin to realize, woo, what we thought was real simple isn't quite so simple. And that's what happened with DNA, of course. They knew that something was going on. They knew that something was being passed on from one person to another. And then when some experiments were done with the blossom of some pea plants many, many years ago, a priest by the name of Mendel was able to demonstrate heritable characteristics that would go from one generation to the next, or it could even jump over a generation and show up two generations later, which we see happen many times in the health field, that problems of the grandparents show up in the grandchildren rather than the children. And so as the genes get in and the pot is stirred and the possibilities come to the surface, sometimes it's a little different than what we would like it to be. So the big thing, of course, was to crack this code. How did it all happen? And then down the road some experiments were done that led to believe that it was the nucleus, and that there was something in the nucleus that was carrying the information. And without going into all the details of how this came about, which is really not pertinent to what we're talking about, it eventually came down to the fact that they knew that there were something called nucleic acids, and that they were somehow involved with the genetic, and it was known that there was some kind of a structure as to how these genetic factors organize in the nucleus of the cell. And so it was, of course, once all the information was there, it was down to a matter of determining the structure. How did these things all fit together? And the big race was on, and it was known that whoever did it was going to receive a Nobel Prize. And so some of the top minds in the world, including Linus Pauling in this country, and two students at Cambridge by the names of Watson and Crick, they were all working on, just how does this structure come together? We know now kind of what the components are, but how do they lock together? And so it was first published, big article in Science Journal by Linus Pauling, who was considered the man who had written a textbook on the nature of the quantum chemical bond. And so everybody kind of thought that he was going to be the guy to break the code, or not to break the code, but to break the structure. And the code came later. So as time went on, bit by bit, after he published, it was of course discovered that his structure was wrong, that it violated some of his own principles of nuclear bonding, or not nuclear, but hydrogen bonds. And so consequently, he kind of embarrassed himself to the world scientific community, a man of his stature violating his own principles of bonding. And at first, when Watson and Crick heard it was published, they were very disturbed because they thought, uh-oh. And then when they read the paper, they said, well, this is crazy. He's violated his own principles of bonding. It could not be correct. And so they were back in the race, and bit by bit, over late night encounters with the different nucleic acids and how they might fit together. It was basically like a jigsaw puzzle of getting it to all fit together. And finally one night, zang, they made the breakthrough, and they rushed right out and published a one-page paper about their findings, which was published in Nature. And lo and behold, they had assembled the correct structure, and now the race was on to understand it. So there's a big difference between knowing the structure, which is how the backbone, which is a combination of phosphorus and sugar, and which is how it got its name, deoxyribonucleic acid. And it's ribose is the sugar that's part of the backbone, along with phosphorus. Out of that, they knew, okay, somehow these links, and what they found out pretty early was that only certain nucleic acids would connect with certain others. And so they found out that adenine would only connect with thymine, and cytosine and guanine would only connect. So C and G, A and T are the combinations that they will connect. Okay, so that was the next big step. We knew how they connected, and that's how this formed this big structure called the DNA molecule, which I'm sure by now, unless you've been totally blind and had your ears plugged, you probably by now have seen numerous times the DNA symbol of the double helix with the steps across. Okay, so but it wasn't that simple. Okay, now we know the structure, but how does it work? How does it make or code for for amino acids that make proteins? How does that happen? See, they already knew it, somehow it had to happen, because these protein molecules would appear in the cytoplasm, and they knew they were being manufactured, and they knew they were being manufactured from the amino acids, but they did not know what triggered and organized it into a chain of amino acids to form a protein. So that was the next big hurdle to come down the pike, but it was around 1961 that a researcher at the National Institute of Health, who was not really a molecular geneticist, but thought that it was an area that needed some investigation, took it on as a project. In about 1961, the first breakthrough to demonstrate that the DNA codes by what are called triplets. In other words, when you look at a picture of it, you'll see that there's groups of 3 steps like a ladder. 3s, and then another 3, and then another 3. Well, each of those groupings are different depending on which side the A's and the T's are, and which side the G's and the C's are, and how many of each of those combinations are in a group of 3. And that group of 3 is the code for one amino acid, with the exception of 2 step and start codes. So, start and stop. So that means that when you want to to code something, a message comes to the DNA. Okay, so you get the message. Now, we're not going to get into the message part of it, and maybe we will a little later, I'm not sure, because now we're talking a whole new ballgame. Because at the beginning, it was believed that somehow in the body it got triggered, and the message went to the DNA to open up a particular gene that had a group of these triplets and open the strand apart into two single strands with half the code. And then there were enzymes that built another half to that exposed code, and that half was RNA. So you have DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, and the RNA is half one of the rung, or the spirals of the helix. Okay, so, but it's enough to carry the information. The only difference was that instead of it being A and T, for some reason, and I'm sure that in the wisdom of nature there was a reason, that in the RNA, instead of T showing up, thymine, it is uracil, a U. And so the U designates what in the genetic code was a T. So anyhow, this pattern or this message that comes from the DNA goes out of the nucleus into the cytoplasm, and there are structures in the cytoplasm called ribosomes. Now these are called organelles, all these little structures inside the cytoplasm of a cell. And so the ribosome takes this strand of RNA, messenger RNA, and picks it up and starts to read it. So it reads the first group of three, the codon, the first codon of 3, and it comes up, okay, this is tryptophan. So the message goes out into the cell, and another type of RNA called transfer RNA picks up that particular amino acid and brings it over to the ribosome, and it locks it in, and then it reads the next triplet for the next amino acid, and again transfer brings it over and it locks in. So amino acid by amino acid, it builds a whole chain to make a protein molecule. Now it, as I say, has a start message and a stop message, so that it just doesn't keep filling the cell full of this protein until it finally overloads and kills the cell. So the start and stop information is there, and it builds that chain of amino acids. Okay, then it releases them, and then a peculiar thing happens. Depending on the sequence of the amino acids, they start attracting each other until pretty soon they form a three-dimensional protein molecule. And the form of that three-dimensional molecule is based on the sequence and the type of amino acids that have been coded into that chain. But, okay, so that's the end of it. We now have information that was stored in the DNA creating a message of RNA, taking it out into the cell cytoplasm, being picked up at the ribosome. The ribosome then attracts the correct amino acids based on the code, and it makes a chain, and when it hits the stop triplet, it stops the process, and now you have this protein molecule. Okay, so, and I said it depends on the nature of it, how it forms into a three-dimensional structure, because I'm sure if you've looked into any biochemistry books or whatever, you're going to see that protein molecules are three-dimensional, rather complex molecules made up of many amino acids, and that the configuration is different depending on the particular protein molecule. And then it gets leaked out into the system, depending on where it needs to go, and it is attracted or directed, probably both, to the right area that is needed to rebuild structure, to build it builds hormones. I mean, there's, hormones is actually a chain of amino acids, and so are enzymes. The way they assemble causes them to be functional in different ways. And so all these amino acids in the body then are used to build all these proteins and to build hormones, and as I say, hormones and so forth. And there's other parts in the hormone, but there are amino acids in there too. Neurotransmitters, all kinds of things in the body to make the body work are made from these amino acid combinations or chains or complexes, whatever you want to call them. And anyhow, that was it. Real simple. Now, they didn't know how the message gets sent. They knew it had to somehow. And they didn't know a lot of things, but the one thing they did believe at that point is that it was fixed. So as soon as this whole idea came together, which is, I said, by the early 60s, actually by 63, they had pretty well mapped a, should say the whole code, the whole basic code. And it can go in a nice chart like this. I don't know if you can see that or not. And so you can look at U, C, A, and G, U, C, A, and G, and as you look out and combine these together, they form the different codes for the amino acids. So phenylalanine, leucine, valine, methionine, tryptophan, arginine, all of them are there. And as you probably noticed, there was some stops in there and so forth. So this was what was discovered from 1961 through 1963, and that was called deciphering the genetic code. And so, of course, the gentleman or gentlemen, in this case there was two people again, were credited with, in fact, I think there ended up four receiving the Nobel Prize before it was all done, on deciphering the code. Okay, so now we had the code, and now we have the structure of the DNA, and we thought, wow, we've got it all made. Now we know there's a gene that codes for everything, and that's it. If that's the genes you have, then that's what you're going to become, and there's not a thing you can do about it. So right away, the pharmaceutical company said, wow, if we can figure out some way to get in there and take out a bad gene and splice in a new gene, we've got a whole new market of very, very expensive testing and very, very expensive drugs. And we're going to open up a whole new scenario for the pharmaceutical market. And wow, it's going to be a big, big money maker, because now who wouldn't pay to have a defective gene replaced by a good functional gene? I mean, wow. Just think of the marketing department of the pharmaceuticals when they considered the implications of this knowledge. And it started them on a project called the Genome Project. You've probably heard of it. And what they said is, what we're going to do, we're going to go through and we're going to actually understand the whole genome of the human gene, and therefore we'll know all of the genes. And therefore, it's a matter of mapping what each gene is for. And pretty soon, as I say, it's going to be a mammoth market of doing testing. And back in its early days, it was thousands of dollars to get some genetic testing done. So it was going to be a whole new market. We're going to make huge amounts of money. And all because we're just going to slice out a gene and put a new one in, and that's it. And if we don't do that, you're stuck. Whatever you got is what it is. Now, out of that grew the whole idea of eugenics. Had started way back, and it got, before they even got down to this point, they were already thinking, okay, we're passing on stuff from one generation to another, and what does that mean? We need to get rid of, or in some way neutralize, those people with defective genes, because if the person is a criminal, then we know their kids are going to be criminals, because it was not only thought that the body structure, but the body behavior or the mind behavior was also affected by the genes. So the thing to do is to get rid of all the defective people. And another major player in the world, Adolf Hitler, had the same idea, him and his party, that they would just wipe out all the defectives so they could not intermix with the purebreds. Very simple thing, and as I said before, Harvard actually in the 30s had a degree in eugenics, which is what it was called. And it was a good idea, and of course once it caught on, all the unfortunate people that were considered derelicts of mankind, thousands and thousands were forcibly sterilized so they could not pass on their defective genes. So it's kind of interesting where things lead and how fast man can jump on a tangent and go way off someplace, and they did. Now, I didn't mention one other part of the genetic factor, and that is the mitochondria in the cell. The cells have low structures, and I thought maybe I do have a photo. I don't know if you can see it or not, but it gives you a rough idea. These little organelles are kind of not exactly oval shaped. They look kind of like a type of, I've seen before, a seed that is actually elongated and rounded on the two ends. And so the mitochondria, when you see actual microphotos of them, that's kind of the shape. But for all practical purposes, an oval shape represents a mitochondria and a mitochondrium. Mitochondria are they're little structures that have a series of folds inside, as you probably noticed if you looked at that pretty close. There's little orange folds, and that's where the respiratory chain ultimately travels back and forth across those walls, moving protons and electrons and creating a type of molecule called an ATP, which is an energy molecule that fuels everything in the body. In other words, they're little body batteries, micro in size, inside the cells, and they store up energy by way of something called high energy phosphate bonds. And ATP means adenine or adenosine triphosphate. And they are made from adenosine diphosphate, which are two phosphates on the adenosine, and then they add a third one, so it comes out to ATP instead of ADP. And so they sit waiting for the need of energy, and when they do, they grab one of these, and when they release that third phosphate bond, the high energy is released, and they have energy to do things. The body does. The cells to rebuild, to function, to carry on everything, and so they're very important. However, it was found again in the 60s that the mitochondria actually had their own DNA. But it was kind of in a ring, and it operates differently in a lot of ways, in ways that we probably are not going to have time to talk about. But they liken it to a loop or a ring. Now, it isn't really that way. That's just for illustration purposes. It's more like a rubber band. In other words, it is a ring, but it's very flexible, and it's not a perfect ring. It's kind of irregular like a rubber band would be, especially a larger rubber band. It kind of can form an irregular pattern when you lay it down. And so that's what the circle of the DNA looks like. Now, that works somewhat different, and it's interesting because it only comes genetically from the mother's side. And, you know, the mitochondria, there's lots of them. Some cells have a few, some have as high as hundreds, and maybe a couple thousand or so. And like the heart muscle, because it needs so much energy, has a huge amount of mitochondria forming energy all the time. So that is a big issue then for the mitochondria to make sure that it can produce all this energy. So that's why I believe that the DNA needed to code for the function of the mitochondria are so important to living and functioning that the nature put the DNA in right where it's needed to make the factors needed to help the mitochondria produce energy, instead of having to wait for messages to get out to other cells in the body, or even the DNA of that particular cell, and bring over the things needed for that to work. They're all self-contained in this little unit, and consequently, because it comes only from the mother, it's a good way of tracking genealogy in terms of determining what your genetic line is, because especially down through the mother. And they use that a lot for identification and several things. It's interesting because in the process of fertilization in the human, and I'm assuming other animals also, the sperm carries half the code for the nucleus of the cell, and the ovum contains the other half, and the two of those joining together form the genetic code for that particular new conception. Well, the interesting thing is the sperm is carrying also mitochondria to make energy, because they have to move to propagate, so to speak, and consequently, when the ovum picks up a sperm, it not only takes out its genetic code, but it kills off all the mitochondria, so whatever, and breaks it down with enzymes is what it does. So consequently, then there's no mitochondrial DNA can be passed on from the father, only the mother. And so it's kind of interesting how nature has eliminated a lot of confusion in that respect by carrying that out. And now there's another aspect, and this came up along later, and this is becoming a big thing now, and that's something called epigenetics, which means around or beyond or above the genes. So there are factors in the body that carry on a lot of processes and that affect the way genes express that really, they're not genetic per se. But the interesting thing, they can get passed on to the next generation, and that can cause an inhibition of certain genes being expressed, or they can encourage a gene to be expressed. And it kind of is, one of the factors involved is something called methylation. A methyl molecule added to a gene blocks its expression, and an acetyl molecule added will trigger expression of a gene. So somehow the epigenetics are able to use that, and probably some other factors, to determine what genes get expressed. But now, getting back to the genome, the Genome Project, they already typed the gene code to the mitochondria because it was very small. There wasn't a lot of genes there. But the big DNA in the major cell, different story. So it took them years to do it, and as I say, they had a reason for doing that. It was going to be magic for a lot of reasons. But bottom line, when you get down to the bottom of the page, it was all profit. We're going to make a fortune off of this. Okay, so they determined that there was probably going to be about 125,000 minimum genes in the human genome. And because they knew there's at least 100,000 proteins in the body that need to be coded. And so there had to be a gene for each of those. And then they also knew that there were controllers and so forth. So they estimated 125,000. Bottom line, when the genome, and they kept talking about it all along, once we get the human genome decoded, we're going to do all these genetic therapies, and we're going to make a fortune, basically, is what they were saying. They were letting the public think, boy, we're going to be able to help you in so many ways, but at a cost. And anyhow, the big shocker came when they finally completed the project. And they found that the human genome had somewhere around 25,000 plus or minus genes, which is about 100,000 short to do what they anticipated it should be there for to do. And that was a shocker. And suddenly the genetic therapies got kind of quiet. Now you still hear some bits and pieces here and there, but all of a sudden they were confused, because then they found that certain things trigger genetic, like the epigenetics, and they found out that sometimes one gene would be triggered, which would trigger 2 or 3 other genes, which in turn would trigger 2 or 3 other genes, which then that's the ones that got expressed. Now, how are you going to inject into that and make a change when you don't even know what the final sequence is that's going to carry out what the project is? In other words, the body is going to do this, and to do this it has to do this and this and this first, and we don't know what is that sequence. It's like rolling dice twice in our head to get a final answer, and therefore it's pretty rough. So it's been relatively quiet. There's still some things here and there pop up. You know, you still hear somebody say a fat gene and a gene for this and a gene for that. Doesn't really come out that way. Now, at the same time, there's a lot of things going on that are really getting into understanding the genes. Now, that's where, when we get into some of this ghost thing is going to come about. The Russians have done some tremendous research, and there's a lot of other stuff going on, and some of the just very recent things that are happening are just outlandish. But the whole thing about genes that I thought was kind of interesting is when you look at some of the things that have popped up. Now, as I say, they've gotten pretty quiet on the genes. The idea we're just going to splice in all these genes. I mean, they're doing some things with genes, trying to create certain products and so forth. But the big problem is that they're making some big boo-boos in the process. Somebody has gone through and actually lined up a few things. And of course, some of this had to do with the money thing, of course. But the central dogma, which is what we basically talked about up to the epigenetics, was what they got together about 1957 and settled even before they knew what the actual codes were. And it was basically that it's all fixed and blah, 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 which we talked about. So, for some time now, there's not a lot of talk about DNA, but they've been doing research on the side, trying to manipulate the genes in some way. So, over the past 50 years, here's some of the things that's going on. We have witnessed the cynical absurdity of patenting isolatable genes as human inventions. In other words, I think I mentioned, in fact I know I did, back along the way, about Henrietta Lake, who was a lady who had cancer and died of cancer, and they took one of the cancer cells, or a group of them I should say, and they started culturing them in the lab, and they have sold them over the years to labs from those cultures. It made millions and millions of dollars. The unfortunate thing was that Henrietta Lake died penniless, and didn't even have money to bury her, and they never got a nickel out of all that. In fact, they didn't know for many years that they were making all this money off of her genetic. And of course, you know, it was a disturbed genetic, and that's why the cancer was there. And it's lived on indefinitely. In fact, there's some other factors, and we don't have time at this point. I've mentioned it some in time past about telomeres. But anyhow, the cancer cells continue to live on and have replicated thousands, if not a million or more times, maybe 2 million. And they just keep going. As long as they get nutrients, they just keep replicating or growing, and they don't die. And so, talk about the immortality factor. Okay, so it says that some of the doctors, and I've known this to be true, some people would go to a doctor, the doctor would want to do some testing, and then he would take the genetic information of that person and go to the U.S. Patent Office and file a patent on it, and then sell it to a university or universities for millions of dollars. And so people, when they found out what was happening, they've sued the doctors, and guess what? The court upheld the doctor. Yeah, sure, they can do it. Talk about disreputable, and talk about money being bottom line. Okay, so we have witnessed a global proliferation of genetically modified foodstuffs. And so you've probably heard a lot about that kicking around, the GMO thing. That's not the subject of today, but the fact is they have done genetic alterations, and again, profit. And when some of the studies were done outside the country, it was showing that they were very, very harmful to other living organisms that had to live on those genetically altered plants. And so they, in this country, the major protective agency of our government has made the comment that we're going to assume that the GMO food is safe, and so they do not require it to be tested. And in those few cases where the industry has done some of their own testing, for some reason it always seems to come out positive as opposed to independent laboratory testing coming out negative. So it makes one begin to wonder about the research industry in general. And also, they've cloned some animals. Everybody knew about Dolly the sheep. Of course, what they don't know is that very rapidly the sheep aged to the age of the sheep that the information was taken from. And number two, they had to euthanize the sheep at age 6 because it was so crippled from arthritis and lung disease and so forth. So the clone didn't turn out so well. And there's a lot of new things coming on that makes me wonder, and we won't even go into that. They've got some pigs that were genetically altered, because they're not only doing it to plants, they're doing it to animals. Some of them have developed arthritis, ulcers, blindness, impotency. Salmon grow 50 times faster than normal salmon, turned cannibalistic because they couldn't get enough feed, and kind of crazy. And some terrible looking stuff. They've genetically produced some chickens that with no feathers, but they can't even stand up. And they can make them grow so fast that from hatching to slaughter time is 42 days. Now that's pretty fast, about 6 weeks roughly. And they look awful. I mean, it has to lay on the ground because it can't walk. They really screwed it up good in the genetic process. So that's some of the things that have been going on. And there are some good things, I have to say. There are some altruistic type researchers out there, but they're not motivated by big money, and they're not being funded by big money. As I've said before, in this country, bulk of the research is money driven, and therefore you don't research things that are humanitarian as much as things that we can push off as being humanitarian, but at a very high price. And so researchers aren't going to research anything that they can't get funding for, and the only funding in most cases that's available is for a company who has something they want to market, and they want some kind of research that they can use to back their product so they can put it on the market. And so that's an entirely different ballgame. And that means that the researcher is not going to be very inclined to go back to the person and say, oh no, we found this stuff is terrible, it's going to kill people, it's no good. Somehow they're going to maneuver the research and tweak it in such a way that at the very worst it doesn't seem too bad, and its best is wow, this is the greatest new thing that's ever happened. So I'm very what do I want to say? I'm not very optimistic about the research that's being carried on currently. Outside of there are, again, as I say, some people that are not money motivated, that are independent researchers, and they're just looking for information, so they have no interest in the outcome either way, other than the knowledge of what goes on. And so that's true research. These people start out with some question, and then they begin to do all the testing, and whichever way that comes out it may not be always the way they kind of would have liked it to have been, but they are willing to stand by what the final results are, so they are much more objective in what they do. Unfortunately, as I say, it's not a tremendous amount, because you have to get private money, because the big money is not going to throw anything into something that's not going to return to them a huge profit. Okay, so I think we're getting kind of down toward the end here of this broadcast. I kind of thought there would be 3, but I wasn't sure. But I've saved some of the most interesting and outlandish things to last, because remember I said that it was from the mechanics, which is what we've pretty much been talking about, to the ghosts. And the next things we will be talking about you're going to find to be pretty fascinating. Some of the Russian research, some of the things that they've learned about the genes. For example, I'll just throw in that the genes are sensitive to, now this is the simple one, the genes are sensitive to grammar, syntax, and words. It was found out that there's a certain syntax and grammar in the way genes organize and work, and that of course they're vibrating at a certain frequency range, which unfortunately happens to be the microwave ranges, somewhere about 54 to 5456 to 7456 gigahertz is the frequency that the DNA vibrates at. And so microwaves happen to fall within that frequency band, and so I believe the genes can be damaged and altered by microwaves. Including food microwaved in a microwave oven. If you eat very much of it, I think it can cause some very adverse effects. On the other hand, if you can take words like to do something, for example, to correct yourself, the DNA to correct a particular problem, if you can modulate a gigahertz carrier wave in that band of frequency vibration, it appears that you can affect the DNA and the expression of the DNA and cause some interesting things to take place. That's a discovery that was made that I think is kind of fascinating, and that's kind of the beginning. But some of the things that are happening now, you're just going to, you think, wow, this must be science fiction, it couldn't be true, but it is true, it's already being done. So we'll be talking about that in our next broadcast, some of the ghostly implications and the things that sound like it couldn't be, like emotions can affect the structure and function of DNA, self-programming can have a big effect. I mean, we're going to talk about several things that is actually being done. It's not just an hypothesis. It is actually being done. So, our time is up. It was nice to be with you again. It will wrap this up the next time on number 3, on the mechanics and the ghost of DNA. It was nice to be with you again. Go to our webcast, mindyourhealthcast.com. As I say, bit by bit, we put different things on, and we're working in the background towards some major revision of all that. And I'll let you know when that's going to happen. Otherwise, it was nice to be with you again, so be healthy.